Next topic will be genetic disorders. Under genetic disorders, we will say about first one, sickle cell anemia. Sickle cell anemia is considered as autosomal recessive disorder. Now, it is also considered as qualitative inheritance or qualitative variations. Qualitative variations. It means that the type of disorder or genetical or gene change, it is not influenced by the surrounding atmospheres. For example, if you take skin color, when you go for the hot sun, and it means your skin is exposed to the hot sun, your skin color will be changed for a while. That can be considered as quantitative inheritance. Means just not because of that, basing on the quantity of the alleles or the types of genetic gene forms, there will be change in the color. Now, in case of sickle cell anemia, which is also called as qualitative variation or inheritance. Inheritance. I already told you qualitative variation. It means that it is not affected by the changed environmental conditions. Now, this sickle cell anemia for the first time was found by Herrick. Now, what causes this sickle cell anemia is due to change in 6th position of the codon in beta chain of hemoglobin. So that what happens? Normally, when there is change in the codon sequence or codon type of beta chain of hemoglobin, we know that hemoglobin is existed inside the RBC. So if this is RBC, RBC shape is biconcave shape. Biconcave shape of RBC. This is normal condition. Because of this change, the shape of RBC is changed to sickle shaped. So that we are giving a term like sickle cell anemia. Coming to the genotypic expression of this sickle cell anemia, genotypic expression can be one X chromosome with normal type of genes that codes for normal healthy hemoglobin. Another type of X chromosome which is nothing but another autosome of the pair, which is also coding for normal alpha chain and one beta chain. We can write here B also. So it can be considered as normal condition. Normal hemoglobin or normal autosomes we can consider. But if one X chromosome is coding for normal alpha chain and the other hemoglobin molecule means beta chain, if there is any change because of the gene sequence at the 6th position, it is considered as heterozygous genotype. We will give here A only just to indicate. But here it means that one hemoglobin molecule is alpha chain, another hemoglobin molecule is beta chain. Now it is considered as heterozygous genotype. If it is this condition, if is in a female, if such a heterozygous condition is in female, then female will be behaved as a carrier, or we are calling female as a carrier. If the same condition is in case of males, male autosomes, then male will be considered as the affected person. If in case of male, then considered as affected person. Affected person. If the condition is both the hemoglobin alpha chain and beta chain, both are having such type of sickle cell anemia coding a type of gene sequence or codon sequence, then person will be dead because of anemia. Person dies soon because of anemia. So all the conditions regarding the sickle cell anemia will be only because of the autosomes. We know that in case of human beings, there are 46 number of chromosomes and 23 pairs of chromosomes. Out of that 23 pairs, 22 pairs are considered as autosomes. One pair is considered as sex chromosome. Next. So this is about sickle cell anemia. Second genetic disorder is hemophilia. Now, hemophilia is considered as X-linked disorder or sex-linked disorder. Means it is mainly due to the defect on the sex chromosome. Next, it is also called as Bleeder's disease or Royal's disease. Actually, disease is the term that we use for either any infectious bacterial type or anything. But here in general, we are calling it as disease. But actually, it's not a disease. It is a disorder that is caused by any defect on the X chromosome of the sex pair. The details about hemophilia are first given by John Otto. And hemophilia are of two types, that is hemophilia A and hemophilia B type. Hemophilia A, it is caused due to non-formation of a blood clotting factor 8. We have 13 number of blood clotting factors. Out of that, if there is any defect in blood clotting factor 8, then it can be considered as hemophilia A. Next, hemophilia 8 is also called as anti-hemophilic globulin. It is considered as a protein. Next, hemophilia B, it is caused due to non-formation of blood clotting factor, that is 9. It is also called as plasma thromboplastic component. So, when these two types of blood clotting factors and their synthesis is affected, then the condition will be hemophilia A and also hemophilia B types. Now, if you see the pedigree analysis of hemophilia, so we can consider this one as pedigree analysis. Whenever we are taking any pedigree analysis chart like this, we should take this one as generation 1. Usually in case of Punnett square and whatever the pathways that we are drawing to represent monohybrid and dihybrid, we used to represent the first one as parent. But here we can consider as parent, but that parent is included under generation 1 and this under generation 2. Now, if there was a condition in which father is normal and mother is a carrier. So carrier in the sense, her X and sex chromosomal pair will be one of the X chromosome will be of affected gene that is responsible for hemophilia. Then how the genotypes are distributed and whether any of the baby is affected or not. If the diagrammatic representation is like this, then it is considered as normal male. If it is shaded, then it is considered as diseased or affected male. If the circle is half shaded, then it is considered as carrier female. This can be written as affected means male sex chromosomes are nothing but hemizygous type, one X chromosome and one Y chromosome. So the X chromosome will be handling that affected hemophilic gene. So that particular male will be affected. Here in case of female, when both X chromosomes are there, if the hemophilic defect gene is present on only one X chromosome, then we can consider such females as carriers. Here, if you observe, the female is a carrier who is having the hemophilic gene and it was transferred to the male. Then such type of inheritance is called as crisscross inheritance. From father to the next generation, it will be transferred to the female. Such type of cross is called as crisscross inheritance. But if a mother is transferring her defected gene to the girl child and girl child is again transferring to the grand girl child, then such type of cross is called as non-crisscross inheritance. But here, such type of transferring from mother to son or father to daughter, such type of inheritance is called as crisscross inheritance. If you see another type of pedigree analysis chart, if a father is deceased and mother is a carrier, then what is happening to the girl child? Condition, if father is affected or deceased and mother is a carrier, then we can frame out if father means affected in the sense we have to shade the square box which is represented for male person. He got married with a carrier, means half shading. So here we have to represent the genotypes like this, and here XX chromosome 1 will be carrier. Then what is happening to the girl child? So one girl, second girl, and a boy. If you want to take out that, instead of writing this one by analysis, let's draw within a short period the Punnett square so that it will be easy for us to identify. So here we are seeing a carrier female, normal male, and affected female or deceased female. Next, again affected male. We can write here carrier means there is no normal girl child, so we have to represent as it can be like this. And affected female, so we have to shade the total circle. We can represent her both X chromosomes are holding the hemophilic gene. Next we are saying one normal male also, so simply we can write like this. Here also if you see, the father's disease is being transferred to the girl child. It means that it is also crisscross inheritance. Such condition is also crisscross inheritance. If a question was asked, we'll write this one also now. Affected male, that is X and Y. Affected male. Okay. If a question was asked, like what percentage of males are deceased? Just if the sentence framing is like this, then we should consider out of the total number of male children, and from that how many male children are affected, we should consider. If we see here, the total number of male children are taken as 100%. Out of that, the normal one is 50% and affected one is 50%. But if the question is asked, what percentage of offsprings are affected, it means then answer again will be different. So if we consider the total number of children, when the question was asked what percentage of the offsprings, then we have to take total number of children as 100%. If not, if the question was asked what percentage of the males, then only males should be taken as 100%. And out of that, affected are 50%. So here, if a question was asked like this, answer will be 50%. Next, we'll see about phenylketonuria. We already have studied this phenylketonuria under example, that is pleiotrophy. If you go back and check the pleiotrophy video, you can understand about this phenylketonuria. In this case of pleiotrophy, what we have read is one gene is regulating or controlling many characters. It is considered as, we have seen sickle cell anemia as autosomal recessive disorder. Next genetic disorder, that is hemophilia, as sex-linked recessive disorder. This phenylketonuria is also again autosomal recessive disorder. Here, there was a gene on chromosome 12 that is coding for an special enzyme, that is phenylalanine hydroxylase. This phenylalanine hydroxylase, it converts phenylalanine type of amino acid into tyrosine. If this enzyme is not coded on the chromosome 12, 12 means out of 22 pairs of autosomes, obviously it will be autosomal chromosome only. When this enzyme is not coded, then phenylalanine will be accumulated more inside the body. Next, it will be now converted into pyruvic acid and then gets accumulated in the brain, which is lethal for the person. A gene that codes for phenylalanine hydroxylase is existed on chromosome 12. Now, if the gene is affected or absent, then phenylalanine is highly accumulated inside body and gets converted into pyruvic acid. That pyruvic acid is holding one of the substance like phenyl, that is phenylpyruvic acid. This gets accumulated in brain and damages it. If we go for the function of this phenylalanine, it gets a convert to phenylalanine to tyrosine. So now the tyrosine synthesis is affected. So this is major function of phenylalanine hydroxylase. Next. So this is about phenylalanine hydroxylase function and also phenylketonuria. Next we will see about cystic fibrosis. Cystic fibrosis, like that of sickle cell anemia or phenylketonuria, it is also called as autosomal recessive disorder. When we want to learn about this disorder, first of all we should learn about one special type of gene and some of the functions that are happening on the plasma membrane with help of channel proteins or carrier proteins. Now, on chromosome 7, there was a gene. That gene is represented with capital alphabets CFTR. Now, what is full form of this CFTR? This is called as Cystic Fibrosis Transmembrane Conductance Regulator. What is function of this gene? Function of this gene is it codes for special type of proteins. Those are termed as carrier proteins or channel proteins. What they will do? We know that in case of plasma membrane, if a plasma membrane is like this, there are some internal proteins, there will be some carrier proteins, there will be some internal proteins, longer one, we also call as tunnels. There are special type of carrier proteins that support in transportation of some of the ions like chloride, sodium, etc. Here are external proteins or extrinsic proteins, we say. These glycerols are attached to phosphate molecules. Now what should happen? These carrier proteins, they should allow the transportation of chloride ions from one end to other end. So that what happens? For suppose this is inside and this is outside of the plasma membrane. Suppose think that. This is allowing the transportation of chloride ions from inside of the cytoplasm to outside the plasma membrane. When one chloride ions are getting transported, along with them water molecules also will get transported, and so that in presence of this one and minerals, the mucus is formed on the outermost layer of the plasma membrane, and it will be thinner only because to absorb some oxygen or to have the lubricating of the movement of the particles which are present between the plasma membrane and in case of plant cell, the cell wall. These carrier proteins will do such type of function which supports in existence of mucous membrane outside the plasma membrane. But when this CFTR gene is absent or when it is affected, then what happens? This channel protein cannot be synthesized. If the channel proteins or these such type of special carrier proteins which are supporting in transportation of chloride ions are absent or not synthesized, then chloride ions cannot move from one side of the plasma membrane to other side, means from inside to outer side. Then what happens? Water molecules also cannot get transported, so that the mucous membrane will become thick and will be dehydrated. So that dehydrated or thick mucous membrane, it leads to a defect in the protein synthesis, and inside the body or inside the plasma membrane, the levels of sodium ions and chloride ions will be very much more. Now, and it leads to the type of accumulation of sodium and chloride ions more and more in the intestine. So, transportation of these chloride ions is associated with another type of ions, that those are sodium ions. They should be balanced because if they are more inside the cytoplasm, they should be exported out of the body. Such type of transportation of sodium and chloride ions is clearly given in NCERT in Transportation in Plants chapter under the subtopic that is SIMPORT. Function is transportation of chloride and sodium ions right here, that's SIMPORT, by SIMPORT along with water molecules and maintaining mucous membrane. Now, if CFTR gene is affected or absent, then chloride and sodium ions. First symptom will be when carrier proteins are not synthesized. And the symport of chloride and sodium ions is stopped or is absent, along with water transport. Then what happens? The outermost layer of the plasma membrane will be very thick. It is considered as the dried condition and dehydrated condition, so that more ions will be accumulated within the body. That is also very harmful for the organism, so that mucus becomes thick and is unfit for excess ion transportation from the body, which is toxic to organism. So here majorly during the transportation through the plasma membrane, this fibrosis, in the sense the required protein that is responsible for ion transfer is affected. Means here proteins are affected. Proteins that are majorly working as carrier proteins or channel proteins, those are majorly affected. So here we are giving a term like fibrosis. Next. So if this is affected, what happens? Then, I told you one example, that is in case of plants, transporting plants chapter. Then what is happening to that of human beings or animals? When intestine is not allowing the transportation of these extra ions from the body, then all the blood and muscles, each and every fluid part or internal visceral organs will be accumulated with this type of ions like chloride and sodium ions, so that the suffocation starts. And also more sodium and chloride ions are majorly increased in the sweat. Okay, so this is major effect of cystic fibrosis. So thalassemia is considered as a quantitative variation. Quantitative variation. Now, it is also considered as autosomal recessive disorder. Thalassemia, it is of two types. First type is thalassemia minor and thalassemia major. There are, we can also use some of the terms like beta thalassemia. What is that? Now I will tell. Thalassemia minor, majorly it is caused by heterozygous type of genes. Simply we can tell thalassemia minor is caused by heterozygous genotype. Means this heterozygous type of genotypes, these are present on chromosome 16, and they are represented as hemoglobin A1 and hemoglobin A2. This heterozygous, in a sense, hemoglobin A1 and hemoglobin A2. Heterozygous type of, here I have written A, it means not homozygous, but among that also there are another type, so that I have represented 1 and 2. These are located on Such type of genotypes are located on chromosome 16. Thalassemia minor, also called as alpha thalassemia. And when we are using this alpha thalassemia, then we can use the same term that is used for heterozygous as alpha thalassemia genes. So we can use a special term for this as alpha thalassemia genes. Now, there is not that much important necessary to learn more than this. Just how many types are there: alpha thalassemia and beta thalassemia. What are other names? Thalassemia minor and thalassemia major. That is only important up to for this NEET exam point of view, because in previous MCQs also they did not ask more than this. So just learn the very, very important 1 or 2 terms regarding these disorders. Second one is thalassemia major, which is also called as beta thalassemia. This thalassemia major, it is also called as Cooler's disease. Now, it is controlled by, or it is due to, only one single type of gene, that is hemoglobin B. We have seen thalassemia minor is due to 2 different genes that are present on chromosome 16. Here also here thalassemia major is due to the effect of gene that is present on chromosome 11. This is one single gene defect, gene on chromosome 11. Now, this thalassemia, it causes abnormal synthesis of hemoglobin, and that finally leads to less number of hemoglobin molecules in the blood, so that oxygen transportation is majorly affected. When hemoglobin is absent, we know that hemoglobin is made up of ferrous molecules and porphyrin rings. When ferrous is absent, that condition leads to anemia, severe anemia. So, thalassemia affects hemoglobin synthesis, that leads to O2 transport and also anemia. This is about thalassemia. Next, we will see about another disorder, that is color blindness. Color blindness is an X-linked recessive disorder. Color blindness, also called as Daltonism, because the person Dalton has first observed and explained about this color blindness. Next, if a person is affected by this disorder, then what colors he cannot see? He cannot see the blue color and the green color. Obviously, he cannot see the remaining colors also. Person cannot see blue and green colors, and so other colors also. Next, we will see about the pedigree analysis of this. If the situation is like father is affected and mother is normal, so in case of pedigree, if father is affected, we should write like this: who married a normal female. Father is affected means the chromosomes we have to represent like this: X and Y, hemizygous condition, and female normal XX sex chromosomes. And they have children. One is normal male, that is with X and Y. Next is carrier female, that is with X and affected color blindness gene. Again, the son is normal. For suppose, sorry, let's write only up to here. And if this woman marries an normal male, then what happens to the next generation? In the next generation, there is a chance for female, male, male, and also female. Okay. If the children are born like this, the unshaded square box indicates normal male and this one normal female. If the square box is shaded like this, then it is considered as affected male. This we indicate with X affected gene and Y chromosome. If we shade half of the circle, this is indicating X and normal X chromosome. Now this female is considered as carrier. Now, okay. So if we write like this, then it is okay. Let's frame out. I will shift this one to that side, so I don't have place. I have written here the normal female. Now this normal female is again married to affected male. Again he is having color blindness here. Then what happens? We can write like this also. Like this we can write. Then what happens to next generation? Next generation, if you draw the Punnett square, carrier female, normal male, affected female, affected male. So here, this condition of affected female will be seen. If a carrier is married to affected male, female, and affected male are also seen. Means we have to shade like this. Okay. We can also tell that here, okay, this is generation 1. This is generation 2 up to here. Okay. So here, when the carriers are getting married to the affected male, then that particular genes are getting transferred, and in case of both male and female, we are able to see such type of disordered condition. Next, we will see about sex-linked inheritance. Few terms about crisscross inheritance and non-crisscross inheritance or linear inheritance. So by this, I have finished mostly the type of genetic disorders that are present in NCERT. Just one or two terms we will see, and then we will close this topic. One term is crisscross inheritance. Till now I have used in hemophilia and also in color blindness type of genetic disorders. What is crisscross inheritance? It means that it is also called as non-linear inheritance. Now, here the possibility condition is transferring of the so-and-so genetic defect from father to child, daughter to next will be grandson. If we write possibility 1, it will be from father, affected father or deceased father to daughter. Now, from daughter to, again, it should be, if it is transferred to the male, then it will be considered as grandson. Now, since the generation 2 is transferring the same disorder to the next generation, generation 2 here is daughter. So such type of means inheritance, we have to give a term here as digynic type of crisscross inheritance, because here father is considered as generation 1, daughter is considered as generation 2, grandson as generation 3. At second generation, daughter is transferring. Now, this is possibility 1. Another possibility is from mother, affected mother or carrier mother. Now, from her to son in F1 generation or generation 2. We can consider this one as or generation 2, because we are taking the parents as generation 1. Now from son to granddaughter, we can write this one as generation 3 or F2 generation. Here also same, we can use this one as F1, this one as F2. Now here the second generation person is male, so we can write this one as diandric. This is crisscross inheritance, possibility 1 and possibility 2. Next is linear inheritance. which is also called as non-crisscross inheritance. Under this, we can study two possibilities. One is holoandric and halogenic. In case, if the father is affected, then the disorder is transferred to son. Again, this is generation, again son only. 1 generation, 2. This one we can also represent as F1 generation. From son again to grandson. This one again male, this is generation 3. This is haloandric. Halogenic, I'm sorry. Gynic means carrier or affected mother, if she is having that condition, or X and X condition. Then to daughter. Same we can apply the terms generation 1 and generation 2 to granddaughter again. So here it is female, transferring to female, transferring to female children. This condition is called as haloandric and halogenic. One more term we will see that we did not see in case of skin color, human skin color. About polygenic inheritance. There I did not mention about one more because in some types of questions, previous practice questions, I have seen a term that is mulatto-man. What does it mean? It means that person with intermediate genotype. Means genotype is like this. Please remember this. Next, phenotype is intermediate skin color. We can also say our skin color is also like this. So this is another important term. Next, I told you about allelic and non-allelic genes. Next, there was a term in AIPMT 2014 paper. One term was given, that is inhibitory genes. Inhibitory genes. What are inhibitory genes? The genes that prevents the expression of other genes of other loci. Genes of the same chromosome, we can see, that inhibit the genes on other loci of same chromosome. Here, example frequently mentioned is pigment genes of rice plant. Here we have to get one doubt. Normally, if the gene on other loci, if it is recessive, means it can be normally dominated by the dominant genes. But the condition is, here genes we have mentioned means dominant genes that are stopping the expression of other type of genes which are showing same type of phenotypic character on other loci of the same. Now here, inhibitory genes. Again, dominant genes only, this should be. Okay. Now I have written here, pigment genes means phenotypically same character. Same color. Okay. So that these are showing one type of color, these are showing another type of color, but both are dominant genes that are present on same chromosome of the rice plant. Then one dominant is suppressing the other dominant on other loci of the same chromosome. Such type of dominating genes are called as inhibitory genes. Now, this is about the genetic disorders.